Hello everyone, welcome back to our lecture series. Today we will be looking at the reactions of esters. In the last class we introduced the esters and looked at the nomenclature. So the basic thing we'll be looking at this in this module is to look at the reactions of esters. Let's take it off from there. And once again, Emeka Benze is speaking. I have a beautiful quote here from someone popular in the medical world, Mr. Bennett Omalo. Actually, his full name is Bennett Omalo. Bennett Omalo is a Nigerian American physician, forensic pathologist, neuropathologist, and professor. In fact, he was the first person to discover and publish findings on chronic traumatic encephalopathy, CTE, in the brain of former American football players. When he did this job, a lot of people did not have confidence in what he did. He stood against the tide and trying to defend himself and then come up with better evidences to prove everyone beyond reasonable doubt. And it's just recently that he was recognized for the work he has done and has been recognized since then by the Board of Physicians, given the highest award when it comes to is it medical doctors. I'm not very familiar with what that award is, but he stood against the tide, being coming from the minority group, Nigeria precisely, and then moving against the tide in a medical field that is very much popularized by the major ethnic groups. He was able to contribute a lot to this world. So Ben Omalo is my hero. And that is why he said this. Look at what he said when he found this out. He still believed strongly in American spirit, because it was that American spirit that gave him the opportunity to make these findings. He says, trust in the great American ingenuity. We can drive more intelligent, more brain-friendly ways. We can play football. In fact, his finding has actually revolutionized the way football is being played and made sure that players, not just the players itself, but the football industry, the American football industry, has found a better ways of protecting the head of players so that they don't get a lot of feed that will actually lead to them suffering from CTE later in life. All right, that is enough of Mr. Ben Omalo. Let us take it off from there immediately. Now, as usual, our learning objectives, there are two learning objectives. We're going to learn how to write the key reactions for the reactions of esters, and then we're going to recognize the structure of phosphate ester. We're going to know what is the importance of this structure of phosphate esters in biological environment. We're going to know what they do. Well, let's take it off immediately from there. The reactions of esters. There are two reactions we're going to be looking at: hydrolysis and saponification. So let's start with hydrolysis. Hydrolysis reaction is just a reaction of ester with water, in which the ester bond is broken, the ester linkage is broken, and resulting to what? Alcohol and carboxylic acid. Remember, when we talked about ester, the esters are formed when a carboxylic acid reacts with an alcohol. It forms ester. So the reverse of that reaction, the reverse of the formation of ester, is the hydrolysis. So hydrolysis is simply what? The reverse of the esterification reaction, breaking it down to produce those ingredients that led to its formation. Now, it's important to state that the enzyme catalyzed hydrolysis of esters in animal fats and vegetable oils are important process. In fact, when we eat lipid foods, those enzymes that break them down, because most of these fats and oil or lipid come in the form of esters, particularly the triglycerols, triacylglycerol, or the triglyceride. Those ester linkage are broken down by the enzyme lipases. So lipases are those enzymes that break down those ester bonds and breaking the ester down to the carboxylic acid, which we call the fatty acid, and the alcohol. And in this case, the alcohol of the triglyceride, the alcohols of the triglyceride are the what? The glycerol. Okay, so this takes place when fat and oil are digested in the human body. Yes, it takes place when fat and other biological organisms. So let's see what is happening here. Look at what is happening. This is an ester, of course. Remember again that this side of ester is contributed by the acid. So the acid is going to be reforming. That's exactly what's happening here. And this side is actually contributed by the alcohol. In what you are trying to make this thing back. So this is the ester bond. Remember, this bond is what is going to be broken. This bond, this is the ester bond. So what are you doing here is that this bond is going to be broken. Remember, water means H and OH. One of these hydrogen will go and join this thing. If you put H in here to form the acid, carboxylic acid. Whereas the remainder of this R will go, let me use a different color, will go and join this OH group to form ROH, which is the alcohol. So it could be an alcohol or the phenol. If it is an open chain, it's an alcohol. If it is an aromatic chain, the benzene ring, it becomes a phenol. So the breaking down of esters will result to carboxylic acid and what? Alcohol. So that is what we talk about when we talk about hydrolysis. This is exactly what is happening. Let's take some reflection questions to drive home this particular understanding. Now, like I told you, this reaction involves, remember, water simply means, anytime we see water, remember that water is always H and OH. And water is used as a machete to break this bond down. That's exactly what is happening. So you're going to be breaking this bond down. One of the hydrogen will come and join this alcohol. So I'm going to have this thing here. I'm going to have what I have here. Now you have, remember, one of these hydrogen will come and join it to become the alcohol plus. Now this is the side that is contributed by the acid. Remember, this side is contributed by the acid. So what you need to do is to do what? Okay. I think I'm going to work this back a little bit. Now remember, if you cut it from here, this one is going to just be the alkyl group. So you need the whole of this to go join to this to become OH at this point. And then this side, which is contributed by this, will now take only one of these guys to join this place to become, let me change my color again, to become, of course, what we have here is, remember, we have this here, C, we have this here, we have this. So you are contributing the whole of the, you are contributing, there's an O here. So it's taking one of these hydrogen to become this. So what you have here is an alcohol plus, this is of course 1, 2, this is ethanoic acid. This is carboxylic acid. You don't need to name it. Carboxylic acid. This is exactly what we have at this point. And then when I go to the second one, the same thing is happening here, the same thing. So you are chopping it from this point. The moment you chop it from this point, the OH group from here will come and, okay, wait, no, I like chopping it. It doesn't really matter, but I like making it this way. Now, the OH group, again, will come and join here to form this alcohol. Look at this alcohol. To form this alcohol. Actually, this is this is isopropyl alcohol. Or you say 2-propanol. Then plus, then this acid. This acid, of course, will now get this hydrogen here to become this. Remember, you have this here already. And then it's going to take, there's O here. Okay, that didn't come out well. There's O here. And now it's going to take one of the hydrogen from here to be this. So you now have your, again, this is alcohol, and this is the carboxylic acid. So every hydrolysis reaction is going to yield carboxylic acid and alcohol. We take another one. Just take one more reflection problem to drive home the understanding of this hydrolysis reaction. Now, again, remember, you can pause this video at this point in time, walk through these problems, and then come back and see how I did it for comprehension. Again, the same thing is going to happen. I'm not going to put my arrow, but you already know what is going on. This is the acid part, so I'm going to put the acid part first. Of course, this doesn't change. Here, we'll now get the OH to become the acid, plus the alcohol part is going to be, remember, this one is here, and then I'm going to put this. This is actually third butyl, and then you're going to have this becomes your acid, carboxylic acid, and this is your alcohol. The same thing goes here. In this case, we have benzoic acid. Remember, this is from benzoic acid now, from the ring. So what it means is we're going to be having a benzoic acid from this point. So break this thing down again. We're going to break it down from here using water. Hydrogen will come and add here. So if I draw my benzene ring, as usual, I'm going to use the shortcut of benzene. You can always put this 0 to show the shortcut. That is very acceptable. And then you're going to have O here. Hydrogen will take, this hydrogen will join this one to become here. So this is actually benzoic acid. This is benzoic acid. That acid didn't come out well. They were like acid. Plus, of course, now the OH group will come and join this one. You have this. So this is just ethanol. This is 1, 2. So this is typically ethanol. This is benzoic acid. So when you break this thing down, of course, if you want to name this, remember how we name esters? You name this side that is contributed alcohol first. This will be ethyl benzoate. So if you break ethyl benzoate together, you're going to form benzoic acid and ethanol. So if you assume you want to name it, this is ethyl benzoate. But that is not what we're doing here. We're just doing the reactions. Now we move to the next stuff, the second reaction, saponification. Saponification reaction simply means process of making soap, soap making. That's actually what it means. This is process of, and this is very important because every day we talk about soap. These days of COVID-19, I wash our hands all the time. So we cannot underestimate the role of soap in our lives. So saponification simply means how making soap. So from ester, how is it made? So reaction of ester with a base, the reaction of ester with a base, particularly an alkaline. Remember, an alkaline is a soluble base to form soap. Of course, how is it done? In the actual way, preparation of soap begins by boiling a triglyceride. In this case, a triglyceride is an ester that has three ester group. Now, what it means is that a glycerol has been esterified to 3-carboxylic acid. That's why we call it triacyl glycerol. All right. So, it's the preparation of soap begins. Preparation of soap begins by boiling triglyceride. If you don't want to call it, you can call it triacyl glycerol. I'm going to show the picture is here. Glycerol with sodium hydroxide or potassium hydroxide, which is actually alkaline hydrolysis. So what they're doing here is that you are using an alkaline to break those ester bonds, those 3 ester bonds, to form glycerol and carboxylic acid salt. Now the Latin word for soap is saponem, which means soap. Now look at the glycerol. Like I told you, a triglyceride, or if you don't want to call it, you can call it or triacyl glycerol, like I wrote here. This is TAG. Means is that look at what is happening here. This is the glycerol backbone. The glycerol has the first ester bond here, 1, another ester bond, and another ester bond. That's what we call it, triacyl glycerol. It's a triester compound because it has 3 ester bond. Because you know, glycerol, look at glycerol, it has 3 hydroxy groups. So each of those hydroxy groups react with, it actually has an ester with an acid. That's exactly what we call it, a triglyceride. So look at what is happening here. In this triglyceride, what happens? Sodium hydroxide provides that machete that comes to break those bonds. Look at the bonds are broken, 1 from here, and from here to form glycerol. So the moment you break it, this, look at what happened. The moment you break it, this OH group will come and join this. So this OH group will come and join at this point to form the 3 what? OH group in each of these 1, 2, 3 carbons to form the glycerol. So anytime you form a soap from glycerol, triglyceride, you're going to be forming glycerol plus the soap. So the soap itself is a carboxylic salt, or you can say carboxylic acid salt. It's a carboxylic acid salt, or you can say it's a carboxylate salt. Of course, in this case, you can call this sodium carboxylate, just as a general name. It's sodium carboxylate as a general name. So it is this sodium carboxylate that is the soap that you use in cleaning your hands or by taking your bath in the bathroom. So in the actual sense, how does soap actually function? In water, what does the soap do? In water, soap molecules spontaneously cluster together into spherical molecules we call the micelles. Now, a micelle is a spherical arrangement, of molecules such that the hydrophobic part of the micelle are protected or shielded from the aqueous water environment. Remember, when you wash with water. So the hydrophobic part, which is usually the tail, is shielded from the aqueous water environment. Whereas the hydrophilic, remember hydrophobic means water fearing, the side that doesn't like water, water fearing. The water fearing part. Oh no, I didn't change that. I wanted to change it. So the hydrophobic is water fearing. It doesn't like water. Whereas the hydrophilic is water loving. So the hydrophilic is water loving. So the water loving part, I don't think that I like how I wrote that there. Let me change it a little bit. So the water loving. So the water loving part are now in contact with the aqueous environment when you're washing. That's exactly. So now look at the structure of this is soap. Remember, this is sodium carboxylate. This is the hydrocarbon chain, which is the non-polar tail. It is non-polar, or you can say hydrophobic. Remember, this is the hydrophobic tail. No, that hydrophobic. Now, this side is the polar head or the hydrophilic. Now, this side is what comes in contact with the water, whereas this side is shielded away from water, forming that. So look at what happens. So several molecules of water does this, remember. Now, this is the head. This is the polar head. Let me show you. So here, this is the polar head. And inside here, this is the tail. So inside here, you have the non-polar tail that is shielded from water. So what happens is that, remember, most of the dirt we use, some of them are grease, particularly organic compounds that are not very soluble in water. So when they are shielded, what you find inside becomes the dirt. The dirt is what you have inside. The dirt now interacts with, this is the non-polar side. It interacts with the tail that is non-polar. So the non-polar interacts with the non-polar. The dirt, remember this is dirt. R, that's R there. I hope that R came out well. This is R. So the dirt is in contact with the non-polar side. Whereas the polar head, which is, this is sodium now. This is the metal. Let me just say sodium ion is in contact with the outside. Now remember, the water is surrounding it. So I'm going to have some molecules of water surrounding the outside. So molecules of water surrounding the outside. Some molecules of water surrounding the outside. So like I said, this is, I labeled it already. This is the sodium ions here. I already have the sodium ion. So this particular arrangement is what we call the what? The micelle. And this is how actually soap dissolves the greases when you are using it either to bath or to wash your dishes. All right, we now go. Okay, in the reflection question 3, we want to use it to drive home our understanding of saponification. The question says, complete the following saponification reaction. As usual, saponification, like I told you, is alkaline hydrolysis. That's what it means. So we're going to be using this alkaline, this base, to break the ester bond from here. And bring this to come and join this guy here. And then this alkyl group side will come and join the OH to form alcohol. So let's write what the product is going to be. I'm going to first of all draw this particular one. Then I'm going to put this carbon and then O. Now the moment this is broken, remember oxygen here develops a negative charge. I told you that. And then remember this one will come and join, the potassium ion will come and join to form this carboxylic acid salt or the carboxylate salt. Plus, then this is 1, 2. This is now going to form just ethanol. So this is the carboxylic acid salt. You say this is the carboxylic acid salt or the carboxylate salt. Whichever one you want to call it, that is fine. Which is the soap. Remember, this is the soap. This is actually the soap itself. Let me mix. So this is soap. And then plus alcohol. Remember, alcohol comes in as a byproduct. The same thing happens here. Let's see what is happening here. Again, here, you're going to be breaking it from here. Here, sodium is involved. So I'm going to redraw this side. I'm going to put this, nothing is happening from this end. I'm going to put this carbon. And then this O here will not be minus. Then coming with sodium plus. Now this side will now form the alcohol. Remember this cyclopropane ring is there, and then I'm going to put the OH from here. Again, this is the soap, which is the carboxylic acid salt, and then this is the alcohol. Yeah, this is how you complete these two reactions. Now we go to the next one. Again, you're free to pause this video, try out these problems yourself, and come back again to check for comprehension. So in reaction 4, the same thing again. In this case, I have a longer chain, a longer chain ester in this case. In fact, this is from this side is C18. This is the stearic side, and then this is about 3 plus 4. This is the alkyl group side. This is about 4. So this will be like from butanol. So if you break that down as usual, using the potassium here, what are we going to form? So we're going to form, of course, I'm going to put this side, nothing happens from this side. Put this CH2 bracket 16, and then I'm going to make this a little bit shorter, and then this here, you're going to put this, then you're breaking it from this side. This is going to be minus, and then you're going to be having your potassium ion plus this one. Remember, if you now break away this one now, you're going to have, I'm going to put it down here, so it's going to be, okay, let me first of all put the alkyl side. If you put the alkyl side, you're going to be having CH2 bracket 3, CH3, and then remember, from here is where you have the OH group attached. So of course, the one you have up here is the soap itself. Remember, this is the soap, which is the carboxylate salt or carboxylic acid salt. And then this is what? The alcohol. Actually, this soap is called, because this is C18, it is actually potassium stearate. Whereas this one is, since this is 4, this is butanol. But I don't expect you to name it. Remember, when we're doing reactions, we're not doing naming. I don't want to test you too objective at the same time to reduce chances of, making my questions very ambiguous. All right. Now the next one, again, what do we have here? This is an ester. Remember, this is an ester again. This is from, of course, this is a benzene ring. This is the alcohol part. We're going to be breaking it down from here with sodium will be acting as the machete in this case. So I'm going to be having this. And then, remember, this side will stay. So what it means is that this side will become the minus. It will come and join the sodium plus. Now, this is the ethanol part. Of course, this is going to be plus. You can make this. This is going to be CH. You can draw it the way it looks like, or it doesn't matter. I can draw it the opposite. Okay, let me make it the way it looks like. So I'm going to say CH2, CH3. You can put the OH here. All right. Or you can also put dash from there. It doesn't matter. Okay. Well, let me see. Let me make it exactly the way it looks like so that I don't confuse you. I like doing that so that, So if you put the OH here, you now put your CH2, you put your CH3. Actually, this is now sodium benzoate. This is actually ethanol. Remember, you form alcohol. The alcohol here is ethanol, whereas this one here is sodium benzoate. This is the salt. This is sodium benzoate, which is the soap. Remember, the soap is a carboxylic acid salt. So that is how you do these two problems. We now go to the next stuff we have. Esters of inorganic acids. Now, esters can form in inorganic acid, not just organic acid as we've been talking about. So alcohols themselves can form esters by reacting with inorganic acids such as sulfuric acid, nitric acid, and phosphoric acid. Now, as a matter of importance, we're going to be illustrating this with phosphoric acid because this is particularly the one we find in the living organism. So look at what is happening here. This is alcohol, of course, having this, reacting with phosphoric acid. Now, if you look at this, now you see that this is the acid part of this phosphoric acid. Now, these two can form the ester. In forming ester, of course, you know what is going to happen. This one will be removed, and this R will come and join at this point to form this. So this is the ester here. We call this the phosphate ester. Whereas this one you kicked out will come and join, this OH group kicked out to come and join with this hydrogen here to form water molecules. So this is what we call the phosphate ester. So like I said, the esters can also form between alcohols and what? Inorganic acid, inorganic or mineral acid. So in this case, we used phosphoric acid for the illustration. All right. So alcohol reacts with phosphoric acid, we form a phosphate ester, giving off water. So this is also an esterification reaction. So esters of inorganic acid continues. So phosphoric acid, as we know, has 3 OH groups. Because it has 3 OH groups, it can form monoester like we just saw in the first one. This is a monoester. So it can also form diester and triester, just like glycerol. So look at what is happening here. It can form, this is the single ester forming from this part. This is a monoester. Here it can form this phosphate monoester. It can also form here phosphate diester. Remember, this is an ester from here. This is another ester from here. So let's say this is the first ester. Then look at the second one. This is the second ester. This is phosphate diester. It can also form phosphotriester. In this case, let me go back again. This is the first ester. Remember, ester is ROO. This is acting as our P now. You know, this in normal ester, this is going to be C, but what we have here is P. So this is the first ester, phosphoester. Now look at it. There's another ester here. Let me go with the yellow. This, if you look at this is the second ester. And where is the third one? Look at the third one here. The third one is this one. And the third ester is this one. So I'm going to use blue. So the third ester is this one as well. So this is a phosphotriester. And these are very, very important in biological system because the hydrolysis of these esters actually releases enough energy to power metabolic reactions. So esters of inorganic acid continue. Now, let's see. You see, mono and diesters are very important biologically. I already said this. Phosphorylation of glucose is the first reaction of glycolysis, which is actually an activation reaction. What happens here is that glucose itself cannot react. Now, but what happens is that the phosphorylation of glucose activates glucose so that the reaction can keep going, so that the subsequent reactions in the glycolytic pathway will keep running. So the importance of this mono and diesters is that they're important in what? Converting previously non-spontaneous reaction, non-spontaneous process to spontaneous processes. That is exactly the importance. And they do this by activating a molecule that will be getting into a particular pathway. So if you look at, let's see what happens here. In this first reaction, actually, glucose, like I told you earlier, when we talked about the aldehyde, when we talked about cyclic hemiacetal, the glucose usually exists as cyclic hemiacetal. Oh, that's wrong. That is actually wrong. So yeah, okay, this is how it looks like. So now I'm drawing the cyclic structure of glucose here. That's what I'm drawing. You don't need to draw this. I just want to show you an illustration of the first reaction of glycolysis. That's exactly what I want to illustrate here. Now, so this is the reaction. In this reaction now, an enzyme involved is the enzyme we call the hexokinase. You remember what enzymes do? Enzymes facilitate organic reactions, biological reactions, by making them to react faster. So now what happens here is that a phosphoric anhydride, in this case, that comes in here is adenosine triphosphate. We're going to see this in the next page. Is now loses one of the phosphate group. It has 3 phosphate group. He loses one of them to become adenosine diphosphate. Both of these are acid anhydrides. We're going to see this in the next. Phosphoric anhydride. We're going to see this in the next page. Now, converting it to, now remember, this is glucose, the cyclic structure of glucose. This is glucose molecule, which ordinarily will not react. But by activation, by adding this phosphate group to it, it forms glucose 6-phosphate. Glucose 6-phosphate means one of the phosphate group from here is now being transferred to this point to form an ester. So this glucose 6-phosphate is an activated molecule, which does what? Which now continues with the rest of the pathway. So the importance of these mono and diesters is that they play an important role in activating non-spontaneous processes to be spontaneous, and so that the reaction will proceed to completion in biological systems. Again, what I have here, the hexokinase, is the enzyme that facilitates this particular reaction. All right, we now go to the phosphoric anhydrides particularly. Now, the two important phosphoric anhydrides are the adenosine diphosphate and adenosine triphosphate. So what are phosphoric anhydrides? Phosphoric anhydrides are phosphate esters that have 2 or 3 phosphate groups that are linked together. Now, adenosine diphosphate and adenosine triphosphate adenosine diphosphate ADP and adenosine triphosphate ATP are 2 important phosphoric anhydrides of great biological importance. They have great biological importance. So look, this is here. This is adenine. Adenine is a nitrogenous base, of course. You should have known this. It's a nitrogenous base now connected to 2 phosphate groups. You see, from here now, you see this is an ester and this is also an ester. So by here you have 2 phosphate groups attached together. This is what we call ADP, adenosine. So let me use, this is adenosine diphosphate. And of course, there's also that when 3 of them are attached, this is the first one is connected with an ester bond to the second one, an ester bond to the third one. This is the adenosine triphosphate. So it is, if you see what I did in the last page, it is adenosine diphosphate that loses one of its phosphate group to this glucose to form glucose 6-phosphate. Whereas what is now remaining becomes ADP. So what it means is that ATP loses one of these to become ADP. And this is very, very important in biological systems. Now, the last thing I'm going to be talking about in this is for you to see the reaction map. Remember, in all the reactions I talked about, I usually end it with a reaction map. So this reaction map, again, is important for you to memorize. Esters will be having 2 reactions we're going to be talking about. The first reaction is reaction with water, which is hydrolysis, whereby an ester reacts with water to form carboxylic acid and alcohol. This is the hydrolysis reaction. In the second reaction, an ester reacts with a base or an alkaline to form carboxylate salt, which is the soap, to form soap, and then alcohol. And this process, this reaction, is actually called what? It's called saponification reaction. Having said that, we've come to the end of this lecture once again. Thank you for listening. Bye.